Okay, here we go. So we start to talk about growth, and I should probably put the PowerPoint up. Okay, so the growth. Before we move on with some of those notions, then you can put it away later. You don't, okay. So this chapter will include, the unit will include 3 chapters. We're going to talk about bacterial growth and conditions in which microbes can grow, which is actually pretty exciting because we have a chance to talk about extremophiles. We're going to talk about the microbial control, and we can relate it to your household experiences. And we're going to talk about antimicrobial drugs, and then we're going to have an exam. So, bacteria, they're used for division. Mitosis, meiosis, what? What do we call this? Nope. Nope. They use binary fission. Why? They're not enucleated. They're prokaryotes. Prokaryotes use binary. I know it doesn't write. Yeah, it takes a while. Binary fission. Which basically means that bacterial cell replicates its DNA, becomes larger, and then the enzyme called, the protein called FTSZ form the Z-ring here at the cleavage furrow, okay? And FTSZ protein is somewhat similar to eukaryotic protein actin. So it controls the cell shape. And by closing that ring, FTSZ separates one mother cell into 2 daughter cells. Daughter cells, are they identical to mothers? Yes. Practically, yes. Unless, well, there probably are some mutations, but on a big scale, they are all identical. Does that make sense to you? Excellent. So with each division, what happens to the number of cells? If you start with 10, after one division, how many are you going to have? You have 10, we have 1 cell. It divides. How many cells you have? 2. Initial one, it's not because it's divided. The second division, 4. The third, 8. Right? So the time that's required, so each division doubles, and the time that's required for the cell to divide to produce the next generation is called, surprisingly, generation time. So generation time for the microbe greatly depends on the conditions. For example, we put microorganisms in the fridge. Well, we put them in the fridge. Those colonies that you got from the environment. If you took a glance on the plates, environmental samples, do you see a huge number of new colonies? No, because in the fridge, they don't really grow. You put them in the incubator, they will take over everything. Does that make sense? pH, temperature, pressure sometimes, concentration of solutes, concentration of nutrients, all of that contributes to the generation time. Does that make sense? Now one of my, traditional critical thinking question. Microbes. Take E. coli. Okay. It can grow on the plate. It can grow in the human intestines. Where is it going to grow faster? In the intestines or on the plate? I see intestines. Make an argument. Why it grows faster in intestines? Why do you think so? It's fine. Why do you think so? More nutrients? It's adapted to that environment. Conditions are regulated. We have 3 arguments. So, arguments so far, more nutrients. We compare intestines and the plate. More nutrients. It's adapted to the environment in the intestines. Conditions are regulated. Where? Anaerobic. So the environment is more favorable to E. coli because of just the way it is there physically. The generation time for E. coli on the plate is 20 minutes. In the intestines, from 2 to 3 hours. So it grows much slower in the intestines. That's experimental data. You don't have to remember the numbers. No worries. But why? Yes. E. coli has to grow in a company of trillions of other microbes. They all fiercely compete for the nutrients. And when I say trillions, I'm talking about trillions of bacterial cells. So they compete for the nutrients. If you think about the concentration of nutrients in the intestine, look, we all know how feces look like. But they accumulate over human feces, but they accumulate over a period of time. We talk about intestines, they're largely empty. The nutrients are spread like a butter on the, they're not like thick in the lumen, they're on the surface of intestinal epithelium. Does that make sense? They're really thin layer. So there's fierce competition. There's immune system, possibly. All kind of crap that we eat that may not be suitable for the microbes. And now plate, you all by yourself. Nutrients. If you have, say, one cell, it is all by itself. All nutrients to its disposal. It can grow and grow and grow. So generally, microbes in the lab grow much better than in the living organism. Okay? Generation time can be very different. 20 minutes for E. coli. Mycobacteria like Mycobacterium leprae in the lab, the generation time may be 2 weeks. The microbiological joke has that impatient people do not work with mycobacteria because they just cannot wait. Okay? You literally, you have to plan experiment. You put your plates in the incubator and you pick them out in months, okay, with colonies that you can work on. It's really a pain in the ass, okay. The fastest, as far as I know, Campylobacter jejuni in the perfect conditions can divide every 13 to 14 minutes. So it's like really fast. So let's go back to this. E. coli, 20 minutes, generation time. You have one cell. And in 20 minutes, how many cells you have? 2. 40. 4 in an hour. 8. In 2 hours. 64. Yeah, 8, 16, 30, 64. And in 3 hours, 512. And you keep going, okay? So let's say in 10 hours, you're going to have 2 in the power of 30 cells, which is roughly about 1 billion. Does that make sense? In 24 hours, am I a moron? Yeah, I think so. Yeah, it's not going to be 2 in the power of 48. It's going to be 2 if the generation time is 20 minutes. 20 minutes. And one cell in 24 hours will produce 2 in the power of 72 cells, which is a lot. I mean, really a lot. If that would go on and on and on, as we just predicted, the microbes would completely take over the Earth. Why it doesn't happen? Why microbes don't take over the Earth? But they generate really pretty fast. Okay, but no way we could hit that amount of biomass. Seriously, in a day, you have a pound. Imagine you have a cow that adds a pound of biomass every day. It would be awesome. You start with a calf, and in a year you have a gigantic bull that it's just the gold mine. Why? What do they run? Something becomes unavailable. Food, yeah. Does that make sense? Nutrients. We ran out of nutrients, yeah. They ran out of nutrients. Does that make sense? So that's pretty simple explanation. They always limited by nutrients. Always. Even in human gut, only on the plate they're not limited by nutrients. And look at this. The plates that you just threw away, even in the plate with the most dense bacterial colony, they didn't start to spill out of the plate and just grow all over the place. Because as soon as they hit the plastic, there are no nutrients. They can't grow there. That makes sense? Can you calculate, can you observe the number of bacterial cells in the certain culture, like a liquid culture, over time? Sure. We can take samples of the culture, okay, with a known volume, like we did, okay, and put it, say, on the agar plate and spread it and count colonies. Make sense? So we can build a growth curve that reflects the dependence between the number of the bacterial cells over time. How number of bacterial cells in the culture changes over time. Does that make sense? We can measure in different ways. We can count the number of cells, or we can measure the optical density. What is optical density of the fluid? Is this fluid, that's water by the way, is this fluid has high optical density? What about this one? It does. What is the difference between, say, okay, beta comparison. It's crystal violet, and this is alcohol. Which fluid has higher optical density? Crystal violet. What does that mean? Not necessarily, no. Optical density. Huh? You can see through this, but not through this. Light. Use the word light. This one can do what with light? Absorb light. This one cannot. Does that make sense? The higher the optical density, the more light will be absorbed by the fluid. Okay? So, if you would look at the liquid culture of the microbe, the more microbes will absorb. Finish the statement for me. Light, more light, right? So you can measure optical density of the culture. Actually, we'll talk about this measurements. It's very, very simple and very convenient. Now, when you count the cells, which cells do you want to count? All of them? I personally want to count live cells. I don't really care about dead ones. Does that make sense? So that actually reflects the growth of the colony, growth of the culture. So let's see what are different stages of bacterial growth curve are. And yes, you do have to know what happens during each stage, and you have to be able to tell me, you know, this is this stage, this is that stage, certain things happen. So we start at the time 0. We inoculate the culture with a small amount of microbes. Remember, you did the plate inoculation when you did the ubiquity or transmissibility. So you do the plate inoculation, you put it there on the bench. And then if somebody took a glance on those plates when you left, did something happen to them? Or did they look pretty much exactly like at the moment you inoculated? Were there any colonies when you left the class? Like in 2-hour span? No. Okay, this is called the lag phase. During the lag phase, there is no detectable growth in the medium. Why? Main reason, either the number is too little, you cannot possibly sample it yet, and also bacteria are stressed. They are placed in the new environment. They experience environmental stress. They don't grow, they don't divide. They need to adjust to the conditions. Does that make sense? And then they start to multiplicate. Okay? And they enter the growth phase. Now, the growth phase, or logarithmic phase, can be plotted, can be put on the graph in 2 ways. One is traditional, using arithmetic scale. So you have time on the x-axis from, say, 0 to 11 hours. And on y-axis, you have number of cells. So this is 100. This little fragment on the bottom is 100. And then this much bigger is from 100 to 1,000. Obviously, from 1,000 to 1,000,000, I wouldn't be able to fit it on this screen. Does that make sense? So eventually, your growth phase, which follows the doubling of cells every, I don't know, 30 minutes, your growth phase will be practically vertical. Okay? And you wouldn't be able to discern smaller changes in the numbers. If you have total number of, I don't know, bless you, you won't be able to actually see the time span of growth. It would just skyrocket. To avoid that, we use so-called semi-logarithmic scale. Instead of using the number of cells, we use a logarithm of a number of cells. What does that mean? What is the logarithm of 10? What's the logarithm of 10? Not really. What's the logarithm of 10? 1. What's the logarithm of 100? 2. Because it's 10 in the power of 2. Logarithm of 1,000. 3. It's 10 in the power of 3. So take the power that you have to take 10 into to get the number. So if you have 1 billion cells, it's a 10 in the power of 9. Logarithm is going to be 9. It kind of stretches your growth curve, makes it linear. Does that make sense? So during the logarithmic phase, logarithmic, sometimes called growth phase, log phase. During the logarithmic phase is the time when cells undergo fission, divide, and nutrients are everywhere. Metabolic wastes do not accumulate really. okay? And cells pretty happy. Eventually, they hit the moment when cells start to die more and more and more. And the number of new cells, the number of cells that die, become equivalent. Okay? Does that make sense? It's like population growth. What happens to the population in, say, countries of Southeast Asia? Does it grow? Does it fall? Like in Malaysia, Indonesia? It grows. A lot of people are born, and much less die. Okay, so population grows. What happens to the population in Europe? It declines because the birth rate, well, they actually keep maintaining the population because of the immigration, but native population declines because the growth rate, the birth rate, is lower than the death rate. That is applied to any population of any living organisms. So in stationary phase, the division and death are equal, roughly equal. Nutrients become less abundant, okay? Wastes start to accumulate. That makes sense? So cells, say, in the intestine. Obviously, we have a lot of bacteria in the intestine. In which stage bacteria in the intestine exist? Stationary. What would be the main argument against the logarithmic stage? That would be a good scientific argument. Really, and that's why a lot of competition, not enough nutrients. So, but actually, imagine if that would be in a logarithmic stage, overnight we would explode. Right? They would would burst with bacteria. Does that make sense? Right? And if you think about this, that's what happens when you have a bacterial infection. So you have microbe that gets into the wound. Your tissues, like muscle tissue, is it sterile? Your muscles under your skin, is it sterile? Sure. So if something gets there, no competition, plenty of nutrients, it starts to grow. It immediately hits the logarithmic stage. That's why you have a disease. Does that make sense? Okay. So stationary phase. What happens after stationary is the death phase or decline phase. Number of cells that die decreases the number of cells that divide or produced by division. And if you measure the number of living cells, that is important, then you will see the decline of the number of living cells. Obviously, in the death stage, nutrients become less and less and less abundant. They're pretty much gone. Wastes accumulate. Okay. Now, in some cases, in the death stage, especially in the death stage, may arise cells that are called persisters. They slow down the metabolism. They don't get into the endospore or anything. They just slow down the metabolism. And that allows them to survive through the scarce times with the scarce nutrients. Does that make sense? Okay. Persisters, the cells with a slow metabolism that generated during stationary and death phase. Now, bacteria in industry, often the metabolic wastes of bacteria is what we actually want. It's the valuable product. And in order to remove those wastes, we use the devices that are called chemostats. On a very basic level, it's a tank. One end of the tank receives nutrients for bacterial growth. And other end of the tank, there is some sort of a system that removes bacterial wastes. So bacteria exist in the chemostat in the log phase. They constantly grow. Does that make sense? They constantly grow. They produce wastes, quote unquote, okay, and use nutrients. In relation to that, last thing, just kind of maybe a little unexpected. What is the alcohol content of wine? Do you know? No, a little bit more. 13, huh? 12, 13, yeah. In that ballpark. Okay. Why? Why 12, 13? Why not 40%? Why we can't get whiskey just by fermenting, I don't know, barley, sour mash? You can just dump all sugar you have. Twelve, 13% alcohol, like ethanol. How do we make wine in the first place? Grapes. What happens to grapes? What? You squish them, and then what happens to squished grapes? Yes, something ferments them. What ferments them? What? Come on! No, what ferments them? Yeast, bacteria, microorganisms. And when they ferment them, wine, during fermentation, what type of fermentation is that? Alcoholic. You get alcohol. What does alcohol do to microorganisms? Kills them. They cannot make vodka because they will die at 40% alcohol. So we have to collect whatever concentration the initial, I don't know if you call it, the mash? Yeah, the mash. You have to collect the mash and you have to distill it to make something stronger. Yes?
What happens when it changes from alcoholic fermentation to acidic fermentation? So like when red wine turns to red wine vinegar, cider turns to like apple cider.
Different population of microbes. Just different species.
By opening it.
You introduce different species, yeah, and some of them prefer like fermentation, alcoholic, mostly anaerobic. Okay, you expose it to the air, you start to oxidize the ethanol into aldehyde and then into the acid. I can tell you more. Have you heard about, just quick thing? First of all, microbiology of wine in France is one of the best funded branches of microbiology. Second, have you heard about things, I'm not really a wine person, so it doesn't really mean anything to me, but like wine of a certain year from the certain slope of some remote hill in France is different from some other. Why? Because different microbes on the grapes and the populations, the microbiome will depend on the temperature, humidity, exposure to the sun. What France now actually tries to do, they try to isolate the perfect starter so they can get starting set of microbes that gives the best wine and then use it like year to year to make it standard. So that's it for today. I'm going to meet you on Monday.